Hello, I'm Colleen Kroeniger from the Department of Nutrition. My lab has a great focus on liver injury and genetics, and the project that I'm proposing for summer research is the role of the macrophage polarization in the development of sepsis. Sepsis kills 1 million newborns globally every year. The causes of neonatal sepsis can be from congenital infection, which is present at birth, and that comes directly from the mother. Early onset of sepsis which is what the research I'm looking at is going to focus on, and that is onset of birth to 1 week. The infection comes from the birth canal, and finally the late onset sepsis, which we're also interested in, and that's beyond a week. It can be from a maternal or external source of the infection. Macrophage polarization is the ability of the macrophage to go to the tissue and either become pro-inflammatory or anti-inflammatory. We call these macrophages M1s versus M2s. They become either pro-inflammatory or anti-inflammatory because they have a polarizing stimulus. So in the case of sepsis, when LPS, which comes from bacteria, that's a protein in the tail of the bacteria, it will bind to a TLR4 receptor and activate a signaling cascade. This then in turn and increases transcription factors that will then produce TNF-alpha, IL-1 beta, IL-6, and so on. These are known to be a source of inflammation, and they attract other cells. This is important. You need to have some inflammation so that you can get rid of the bacteria through phagocytosis. You also have the ability to get rid of the bacteria as well. In contrast, you want to resolve those M1 to M2. You want to resolve that inflammation, and you do so by activating what's called the M2 macrophage. This polarizing stimulus is from IL-4, IL-13, and so on. This one has very low phagocytic activity, and it basically resolves and remodels the cell and shuts off the pro-inflammatory response. The reason we're interested in it, going back to the sepsis, is that there's evidence that newborns who are very severely ill with sepsis, their macrophage polarization converts from that M2 to the, converts from the M1 to the M2. So to find genetic models that are resistant to sepsis, we are going to use two techniques, and these will be in mouse models. The techniques are called, the first one is a cecal ligation and puncture, and this is a way to induce sepsis in the abdomen of the mouse. So if you look on the left, you can see that there's a cecum and ileum. You have the colon that is ascending as well. If you look at the picture on the right, there's a line going right below the ileum, and that's where it's going to be ligated with a suture. So you're going to tie that suture, not completely off, but you're going to impede the flow. You take a needle and puncture it in that region, and some of that contents from the intestine can ooze out of the intestine and cause infection in the abdomen. You do this as a model of sepsis, and you basically are looking for viability of that mouse model after they have the induction of sepsis. So we will test a wild-type mouse, and we will also test a genetic model. The second is LPS-induced sepsis, and what that means, LPS, as I said, is a protein in the tail of bacteria. It's the signal that there's some bacteria invading, and we need to get rid of it. So you can see in this diagram here, it's binding to TLR4, it goes through a cascade, activates NF-kappa beta, and these are the pro-inflammatory genes that I was talking about, TNF-alpha, IL-1 beta, and so on. What happens is you give this LPS and you measure these production of cytokines, and you look, because all these cytokines are involved in the induction of sepsis. We would also be interested to see if we could prevent that M1 going to M2, because if we could that prevent the sepsis in the newborn infant? So we have a mouse model where we knocked out the PepsiK gene. This is an example of what PepsiK does in a hepatocyte, a liver cell. It can take amino acids such as glutamine or alanine, And those carbons can then enter the TCA cycle, and they can actually leave. Pepsi K is required for the conversion of oxaloacetate to phosphoenolpyruvate. These carbons eventually become glucose, and that's released into the blood. What we did is we knocked out this Pepsi K, but in the macrophage cells. So that is one of the immune cells. We did this through a Cre recombinase technique. On the left, you can see this is looking at, so the white boxes are the control mice, and the black boxes are what we call the macrophage knockout. You can see that when we knock out Pepsi K in those macrophage, that we really have a much more M1 pro-inflammatory phenotype that produces much more TNF-alpha, IL-1 beta, and IL-6. To confirm that it was Pepsi K doing this, we also then took raw cells, which is a stable cell line for macrophage, and we overexpressed Pepsi K. When we did that, the same transcription factors that were elevated when we knocked it out are now repressed. Thus, we suggest that Pepsi K is really important in this regulation between the more M1-like in the knockout and the M2-like in the overexpression. So we will use that genetic model, and as a student in my lab, you will learn some basic molecular biology techniques such as genotyping, RNA and DNA isolation, Western blot analysis, ELISAs. We use the bone marrow-derived macrophage, and we isolate the macrophages from the bone marrow. That's the data I showed you in the previous slide. We'll also do some tissue culture techniques as well. So you will participate with as a member of the Mouse Phenotyping Center staff, Lang Wang, and develop these sepsis models, and we will analyze whether the genetic model that we've developed, is it resistant to sepsis. We will have some lab discussions, probably every other week, where we'll assign a paper and we'll have a group discussion to increase our knowledge about the metabolism in these macrophages, as well as some of the immunology and genetics. By the end of the summer, you will have an increased knowledge of a lot of pathways, and you will be encouraged to write an abstract for the Research Showcase Day. My philosophy about summer research is that I want you to learn a lot, but I also want you to enjoy it. And so we do have a lot of fun during the summer. We'll go out for lunches and such like that. So I hope you choose my project, and I look forward to meeting you. Thank you.